got a broad understanding of the macroscopic organization, microscopic organization of the brain. It's time to put it under the microscope, and let's talk a little bit about imaging, okay? And so now you already, from what I've told you about the way that we have the electrical communication between neurons and then the chemical communication, their metabolic activity, you're ready to understand all right now sort of a broad division in the types of imaging or the non-invasive measures that we can make of the brain, okay? So broadly speaking, we can divide these sorts of measurements into electrical measurements and so-called metabolic measurements. So just briefly, electrical imaging includes things like EEG, electroencephalography, and MEG, magnetoencephalography. And these are measures that reflect cortical electrical activity, right? MEG technically is measuring a perturbation of magnetic field that's done by electricity, but to a first approximation, these are techniques that can measure the consequences of the electrical depolarization of sets of neurons, okay? And so these are techniques that for EEG just involve placing a bunch of electrodes across the surface of the scalp, like in a little cap. And so this is something where it's easy to deploy and it's portable. Someone could wear one of these things around. And another technology called MEG, which has some advantages over EEG, but one big disadvantage is that it's incredibly expensive, requires superconducting detectors, which are cooled by liquid helium, and you have to sit in this enormous thing that looks like a gigantic hairdryer. So these are two techniques which can be used to measure the electrical depolarization of populations of nerve cells. They have the property of having great resolution in time. You can measure sort of by moment to moment how electrical activity rises and falls in an area of the brain. However, they're not very precise about exactly where that change is taking place.
It's a little bit like no smart meters here. This is not telling you what individual houses are doing, but it is telling you that this neighborhood just turned on the electricity. Is that a fair?
Yeah, no, fair enough. That's right. And so the kinds of data that come out of these techniques include plots where across time you're able to show what the electrical activity was. And so you can't quite read the scale here, but this is in milliseconds, right? So down to thousandths of a second, how is electricity rising or falling in different areas of the brain, okay? So you get very high detailed images of changes over time. However, the images over space, this is now a cartoon of the top view of the brain, that's kind of fuzzy looking, okay? Now that's, there are ways of making it better, and people who do EEG and MEG would plead their case and tell you about all sorts of ways they can do better, but broadly speaking, good in time, not so good in space, yes.
How are these most useful?
Well, let me just say from diagnostics, EEG has a long history and got its start as a clinical tool to detect seizures, to be able to tell the different stages of sleep, right? So the way we know about the different stages of sleep comes from measuring electrical activity and its changes. This is how we detect and diagnose seizures. And then from a research standpoint, it's contributed enormously to our understanding of how information is accumulated and processed over time, and how information might traverse from one area of the brain to another during the course of making a decision or perceiving a stimulus. Yeah?
Is this measuring then the axon?
I wish it were that easy. It's actually measuring the depolarization of populations of neurons on the cortical sheet. So it's not as easy as just measuring along those axons. It's actually measuring a spread of activity going across the cortex.
And it's typically the outer regions of the bark rather than the inner regions, because you're coming in. You're not getting to individual neurons. You're just basically at this secondary level through the skull, measuring electricity that gets there.
That's actually a great point. This is that you're putting wires on the surface of the head. You can measure the cortex. If your goal was to see what's going on in the amygdala, remember that was a structure that was way down inside. Not such a good technique. Okay. All right, so now we can contrast these electrical imaging techniques with broadly what I'm going to call metabolic imaging techniques, okay? And so some methods that fall in this category include PET, positron emission tomography, SPECT, single photon emission computed tomography, and fMRI, functional magnetic resonance imaging. And that's the one we're going to talk about some more in just a minute, okay?
And tomography is a $10 word for...
Tomography. Now this was worth a Nobel Prize. This is the idea that by measuring so good old-fashioned x-rays, right? You kind of stand in front of the thing, and you get like a plain flat film of the chest, right? Compare that to a CT scan, right, where you know that you can get this three-dimensional image. The mathematical technique that lets you go from like just a simple plain flat film x-ray into that three-dimensional view of a CAT scan is tomography, which basically involves taking the X-ray emitter source and the receiver and rotating it around the sample, the person. So you just kind of rotate that around, and then mathematically you can reconstruct from what you measured at each angle what the structure of the image in space must have been underneath, okay? So that's computed tomography. And so PET and SPECT make use of that to sort of generate the images that they have. That's why a PET image like this actually has a two-dimensional or three-dimensional structure where you can now, this is a horizontal or axial slice through the brain. That's the back, that's the front. And the relative color intensity here is the amount of signal that was received for whatever was being measured by the PET scanner, which I'll say some more about in a moment. Generally speaking, these metabolic imaging techniques have poor resolution in time, right? They can only measure changes on the order of seconds, right? But they have good resolution in space, certainly better than EEG and MEG. Questions about that so far?
Not at all be pertinent. The limitations, the trade-off between accuracy and time and space, that at all is just an outpouring of the physical limitations, or is it the engineering? What is causing that?
It is not uncertainty principle at all. It is just the fact that the measurements that the phenomena that this measures develops slowly, a little more slowly. The electrical stuff happens with relatively bright lines. Here, this is the metabolic reactions in your brain, and it just doesn't have that kind of same sharp beginning and ending. You know, it's a, and then it's done kind of process.
Right. So, and again, this isn't precisely right, but I think this is a worthwhile level of understanding. You remember those electrical impulses, zoom, they come right down the axon. Okay, that's a sort of a very rapid change. The metabolic things we're measuring, oh, the transfer of neurotransmitter, the neuron factory getting pumping again to cook up some more glucose and consume it, that stuff takes longer and lags behind the very rapid electrical activity that moves along the axons. The first approximation, that's how we can understand the difference in the timing aspects of these two things.
Do that X-ray demonstration, and that was
I guess if I let me do it. Not bad, huh?
You know the airport scan, where you stand and you do this and it goes right. That is I believe using sound waves, but the tomography, the three-dimensionalization.
If only it used sound waves.
Oh,
Actually,
it
yeah, it's
is it X-rays?
Low energy X-ray particles.
Okay,
yes.
so you're getting a little X-ray each time, but it is essentially doing a low energy X. That is the reason that you stand there and it goes around in front of you. Is it's doing that 3D sweep of you, so it can identify location.
So just a word more about PET, because we're about to now turn to fMRI and talk only about fMRI for the rest of our time together this morning. But a couple words about PET. PET turns out to be a really important technique in clinical applications of imaging, and it comes up again in many different forms. Basically, the core way that PET imaging works, positron emission tomography works, is you take a radioactive particle, right? You take something that's capable of undergoing radioactive decay, and you stick it on something else that's interesting to the nervous system, okay? So for example, you could take a bit of radioactivity and stick it onto a sugar molecule, glucose, okay? And then inject that into the blood, and then that glucose, that radioactive glucose, makes its way up into the brain, and then all these nerve cells, which are hungry and munching on the glucose, take up that radioactive glucose and consume it. And then as those radioactive particles decay, they release particles that are detectable by the PET scanner, okay? So if you take radioactivity and you stick it onto some sugar, some glucose, and you inject that, you can make an image of the brain where the signal that you measure at each point is how much glucose was being consumed, right? That might be a very interesting thing to a neuroscientist to know. Or you could basically attach that radioactivity onto water and inject it. And then you find out for each point in the brain how much blood is flowing to that area of the brain. Or, and then this gets really cool, say you have a neurotransmitter you're interested in, like dopamine, right? Now there's a neurotransmitter that, you know, as you'll hear about, does a lot of interesting things. You take dopamine and you stick some radioactivity onto that. And now you inject that in and allow it to arrive in the central nervous system. Now you have a view, an image of where dopamine is being caught by those receptors. Okay?
This is partly how we've arrived at that understanding of the distribution of the different neurotransmitter activity across different regions of the brain, is because we've been able to map it through PET-style experiments, which will tell us where's this stuff going, where's it being caught onto, where's it being active.
Okay. So, but now we're going to leave behind PET and turn to bold fMRI, the nitty-gritty. Okay, so now we're going to really get into this preeminent technique for measuring non-invasively neural activity in human subjects, okay? And the first thing I want to cover is how we get from neural activity, the spiking electrical activity of neurons, to a picture of the brain, okay? So the first thing is you're all likely familiar with MRI scanners as being used as a clinical or medical technique to measure the structure of the brain, right? So these are 3 views of the structure of the brain viewed from the side, or called a sagittal image. It's pretty easy to recognize the structures there. A coronal or front-back image, and then the axial top-bottom, okay? So MRI scanners can be used to get these images of basically the structure of the brain. What we're going to, oh, and it turns out that MRI scanners can be used to get lots of different types of structural images. Here are just some examples. There are different sorts of images which are sensitive to the location of blood vessels. You can map out the blood vessels in the brain. You can run the MRI scanner to get images which show damage to the white matter in diseases like multiple sclerosis, right? A particular type of imaging sequence can do that. You can get a type of image which can show you where strokes are located or where there's bleeding. And you can get a type of image that can be used to trace the organization of those white matter tracts and how they're structured. So there's many different ways of obtaining anatomical information about the brain using an MRI scanner. But again, what motivates the rest of the discussion we're going to have now is the ability to use an MRI scanner to measure something about brain activity, right? Nervous system function. And it turns out the key thing that makes that all possible is something called neurovascular coupling. If we didn't have this, we'd mostly be sunk. We'd figure out some other way around it. But boy, does this help. Neurovascular coupling. So neurovascular coupling describes the following phenomenon. When areas of the brain are more active, more blood flows to it. Okay? Sounds like it's a really simple principle. If you have a patch of cortex, it increases its level of activity, then the local blood vessels increase their diameter, and more blood flows up to that spot.
And you see this in muscular activity as well. If you work out, your muscles pump up. The reason they're doing that is they're getting engorged by extra blood to give more fuel and replenish the glucose and oxygen needs of the muscle.
Right. So that's right. So and it turns out that this is a principle of the adult human brain as well. Exactly how this neurovascular coupling is happening, what it's for, how it varies in different at different ages and in different species is a huge and complicated topic. But just for now, we just need to know that it exists, okay? An increase in brain activity at a local site increases local blood flow, okay? And a consequence of all that is that changes the local amount of oxygen, right? So if you increase the blood flow to an area of the brain, more fresh oxygen comes on in. So what's another, and the next sort of fortuitous property of nature that makes all this work is that the amount of oxygen in the blood alters the magnetic properties of iron. Okay, so now you know blood contains red blood cells, right? Red blood cells contain a protein called hemoglobin. Hemoglobin is the protein that grabs onto oxygen as the blood passes through the lungs. Hemoglobin contains an iron atom, okay, and that's actually what the oxygen binds onto.
And why blood is red, quite frankly.
And why blood is red is exactly right. So it turns out, and so Linus Pauling, right, who you might remember as a guy who won a couple of Nobel Prizes, one in peace, one in chemistry or physics, I can't recall, he first observed that the magnetic properties of iron, of hemoglobin, changes depending upon whether it's bound or unbound to oxygen. So if there's more oxygen, then the hemoglobin has a slightly different magnetic property than if it's not bound to oxygen, okay? So an MRI scanner is basically a long, long coil of wire through which we run electricity. And you might remember from high school physics, hold on just a quarter second, that generates a very strong magnetic field, okay? Leaving aside all the details of the physics, it's possible to use the MRI scanner to measure how much that iron is warping and distorting the magnetic field. And the amount of warping and distorting of the magnetic field varies depending upon whether or not that iron is bound to oxygen. And that in turn varies depending upon whether or not the blood vessels are big or small, which depends upon whether or not there's brain activity at that location or not. So putting that all together, there's a chain of actions that go from a change in brain activity, of cortical nervous system activity, which leads to changes in blood flow, which alters the local oxygen, which changes the iron on hemoglobin, which changes some funky physics stuff about the scanner, which ends up with an image that is, which reflects the amount of neural activity. Okay? So there's this whole chain of events that leads from a change in neuronal activity to a picture of the brain that reflects that activity, right? Which is based upon ultimately a change in blood flow and oxygenation. Let me pause there.
A little more detail or more precision.
Oh, that's such a great question. Okay, so the question is, all right, so I just used a very generic term, activity. And I must say that people who work in this field, who do neuroimaging work, are guilty generally of a very vague terminology about what exactly it is about the property of nervous system function that is driving the signal that we measure. So again, like everything else I've covered so far, this is a very deep and detailed area. But broadly speaking, the type of activity that seems to drive the signal that we measure is the synchronous release of neurotransmitter, right? So what will happen is that if you have a whole bunch of neurons together releasing their neurotransmitter, releasing vesicles simultaneously, that's the aspect of nervous system function that most seems to drive this signal, okay? Turns out that under most circumstances, that's also highly related to the spiking action potential activity of neurons too, but if you really want to know, that's probably the thing that's driving it. Okay?
And that could be both excitatory and inhibitory.
That's right. So now coming back to what we talked about before, neurotransmitters and the receptors can be excitatory and inhibitory. If you're squirting off that neurotransmitter, you might, to a first approximation, not know if the activity you measure is increasing or decreasing the level of activity at the neurons that receive it, right? And again, this is a very broad overview of a very complicated topic. So at the end of the day then, what is produced by the MRI scanner is an image, right? So this is now an axial horizontal image of the brain. Back is back here, front is up there. This is a bit of an older image, so it's fuzzier. They look a little bit better nowadays. But what I mainly want you to appreciate is that a given image of brain activity starts with one of these, which is kind of this fuzzy picture of a measure which is related to the amount of neuronal activity at that point in the brain at that point in time, okay? And this picture is composed, of lots of little points. So it's made up of tiny little points, which we term voxels. It's kind of like if you get in really close to your TV screen, you can see the tiny dots that make up the picture. In the same way, if you get really close into this picture, you'll see the tiny dots that make it up. That fundamental unit is referred to as a voxel. Generally speaking, a voxel is about 3 millimeters by 3 millimeters by 3 millimeters, generally speaking. And I just wanted to give you a sense of what that looks like. So that's 3 millimeters on the tip of a finger. Okay, so that's the kind of resolution that we're talking about. Turns out you can run the scanner to get those the images even with tinier little pictures that make it up. Okay, so that's a 3 millimeter roughly voxel size. And inside that, if you do the math, you come up with about 630,000 neurons inside a 3 by 3 by 3 spot. Okay.
So go back to the,
This one.
does that tell you anything?
Does this one image by itself tell you anything? Not much.
Only a little.
Yeah, not much. Really, where things start to come together is now when you get a lot of those pictures over time. Okay? So you start getting one image after the next over a 2 to 3 second period. And now you're going to look for how nervous system activity, this brain activity, rises and falls over time in response to something that you do to the subject. Okay? And now we finally come to doing something to the subject and making a claim about brain activity. Okay, and that's how we go from raw data over time to getting a processed result, right? A picture of the brain with a lump of activity on it. Okay? So let's go back to this claim. This is the claim. Remember this guy? Brian Dugan. So the claim was that functional MRI can show that Brian Dugan is a psychopath, right? So that claim is based on a more basic or entry-level claim, which is that psychopaths generally show reduced responses to emotional stimuli within the amygdala. That's the core neuroscientific claim that underlies ultimately what's going to be a claim about that guy, Brian Dugan.
What
Okay?
is that based on?
That's what we're now going to unpackage. Okay?
And note that this has some correspondence to the what I was saying about the law and the application is that we've got general conclusions about things, and then we've got specific individual conclusions about things, and these are going to need to map onto each other. The same is true in science.
That's right. So now let's tear this claim apart. Right? Let's understand exactly how it was that an experiment was done that ended up with this as a claim. Okay? So psychopaths. So we're going to have to find a group of people who we're going to call psychopaths. show reduced responses, so some decreased brain response to emotional stimuli, so we've got to figure out what emotional stimuli are, within the amygdala. And as you recall, the amygdala is the structure in the brain that we think has something to do with emotion, okay?
And by the way, we get to the definition of psychopaths typically through some behavioral set of measures. This is this back and forth between the behavioral and the not and the
Which we're going to, which is a key bit of foreshadowing there. Okay, so that's right. So what we do is we put somebody in the scanner. We get a whole bunch of these pictures, which have these individual little voxels. And so when the subject is, so we take a person. Let's just start with one person, and we put them in the scanner. And we get a picture of the brain, like a three-dimensional picture of the brain. I'm only showing one two-dimensional slice because I only have a 2D screen. But it's a three-dimensional picture of the brain, which is made up of these tiny little dots called voxels. And so we put them into the scanner, and we have them look at something. And let's suppose in this experiment, and by the way, this is a study of Kent Keel's, which I'm now going to caricature. Okay, so this is my bad version of Kent's good study. So you put somebody in the scanner, and what you're going to do is you're going to have them look at pictures of faces, okay, over time. So for 30 seconds, they're going to see pictures of people who have a fearful expression, okay? That's a face with an emotional content. And then for 30 seconds, they're going to see a face which has a neutral expression. I think this guy looks kind of angry, actually, but theoretically a neutral expression, okay?
By the way, there's a whole science of developing the suites of these things to present that has been tested.
It goes on and on and on, right? So, okay, so the person's in the scanner, beep, 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 the scanner's running, and they're looking at a screen, fearful faces, emotional faces, neutral faces, fearful, neutral, fearful, neutral. And what you're interested in finding is a point in the brain where over time the signal goes up for the fearful or the emotional faces and comes back down for the neutral faces and goes up and then down and then up, okay? So if you observed this signal over time from this point in the brain in response to these pictures, you'd say, huh, that looks like pretty good evidence that when the fearful or emotional faces are on the screen, there is increased neural activity at that site, okay?
For a normal person.
For a normal person.
Well, for the person you've been
That's
measuring.
exactly.
Yeah.
For the person we've got in the scanner. Okay. So now there's that whole data that's over time. Somebody might do this for an hour in the scanner. Beep, 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 like look at these pictures. You then want to summarize that measurement over an hour. And so the way you summarize it is you say, okay, what was the average brain response to all of the emotional pictures, and what was the average brain response to all of the neutral pictures, right? And you say, well, then how big was that difference, or how statistically reliable was that difference, okay? And you assign a score to it. You can say, well, either it was a small effect, or it was a big effect. You put it on some scale, okay? And then at that point in the brain from which you took this one particular measurement, you could write a number down. You could say, oh, well, it was like a 20% effect, or it was a very statistically significant effect at that point, right? But that's just one point, and if you write the number down, that's hard to see. So what people in this field typically will do is they'll color code it, right? They'll say, well, we'll make a small effect red, and we'll make a big effect yellow, and we'll turn
Make no effect blue.
Sure,
Yep.
and then we'll put that color at that point to indicate how much of an effect there was. Well, that's just one point, right? So boof, we need to do that for the whole brain, right? So every point gets assigned a value of how big the effect was of the emotional face versus the neutral face. And each point then gets a color. And then we get into the realm of what we'll call neuroaesthetics, okay? So this is what the data starts off looking like. At each point in the brain, in this coarse grid of samples, you have a value of the effect. Now, some of those values that you assign, was not a big effect, right? You might say, oh, well, there really wasn't much of an effect there at all. Every point in the brain will have a value, but some values aren't important. So let's apply a threshold. Let's get rid of all the points which are below a certain threshold. And I want to say for each of these steps, there are very good and well-motivated statistical reasons for doing each of these things, right? So there are entirely justifiable reasons for adopting these thresholds. So you say, okay, well, look, just show me those points where there was a big effect or a significant effect, all right? Now, the next problem is that these effects, it turns out that if I apply a little bit of a digital smoothing to this data, I will enhance my ability to detect certain kinds of effects. I'll make it a little bit easier to detect the big things. So I'm going to take this data and apply some digital smoothing to it, okay? Now, and again, turns out there are perfectly rational and well-justified statistical reasons for doing this digital smoothing, but it has the property of enhancing larger areas of response and eliminating smaller areas of response, okay? And again, this is a trade-off that the scientists who do this work know, and they're happy to take it because they do better at detecting the big stuff, okay? You know, though, now we've got this blob, and that looks pretty good, but it's hard to know where in the brain that is on top of this fuzzy picture of the brain. Oh, wait, I know. When I ran the scanner, I also got that high-resolution anatomical picture of the brain, too. How about we show that blob on top of that? Oh, that's much better. Okay, so now what I'm going to do is I'm going to take that little blob of colored activity, of statistical significance, and display that on top of the high-resolution part of the brain. Oh, good. Now I can see where that activity is. You know, but the problem, though, is that this is just one slice. And remember, this is like a big volume through the whole brain. I could just like make a little picture where I put down each slice next to one another, but that's kind of ugly. How about I get my friends over in like the computer science department to make me a big 3D rendered version of it? Oh, that's much better, right? So now I can see the entire surface and structure of the brain. I cut out a piece. I can see the blob of activity. Perfect. So it turns out that every step along the way, these were perfectly valid, reasonable approaches to processing the data, but just appreciate this is a very long way away from what the source data was like, okay? There's an additional challenge. This was the data from just one person. Suppose I have data now from 10 people who I've labeled as normal control subjects and 10 people who I have behaviorally identified as being psychopaths. Well, I need to combine their data somehow. So what I have to do is do brain registration. Everybody's brain is a slightly different shape. They have different patterns of activity. What I'm going to do is take each brain and warp it to a common brain atlas and then average across the subjects. And then I end up with something like this. So now I've got data from 10 people, all their brains warped to kind of make them look all about the same. And I average the responses from all 10. So at the end of the day then, after getting this kind of measurement from 10 control subjects, like normal people, and 10 people who I think are psychopaths, I end up with this. So the claim again, psychopaths show reduced responses to emotional stimuli within the amygdala. What that actually looks like is we're showing these fearful and the neutral faces, and I'm measuring then from within a spot in the brain, say in the amygdala, this was the average response of that area of the brain across 10 psychopaths once I've warped all their brains into a common space, and 10 normal people after I've warped their brains into the same common space. And what I find is that, oh yeah, you know what? It really did work out. There was a larger response to the emotional faces in the normal people compared to the psychopaths.
Although you're going to explain this, the error bar here in a moment.
Exactly. Now, of course, not everybody was the same, right? So some normal people had a slightly bigger response, some people had a slightly smaller response. There's individual differences. There's noise in these measurements. So there are some error bars, of course, on these measurements, right? But I would have to say, to a first approximation, if these were the data and those were the error bars with standard deviations and all the rest, we might say, yeah, you know what? The evidence is pretty good. Psychopaths have a different response from a population of normal control people for this, okay? That they don't respond as much to the emotional faces as compared to the neutral faces.
And that maps onto some behavioral expectations. One of the things that psychopaths tend to do, and in fact part of their diagnostic, is that they have lower affect in when faced with certain kinds of
That's right.
stimuli.
Yeah, so you might say, okay, that's pretty good. But now just imagine or just think about the inferential distance between that claim and this claim, which is this guy is a psychopath because he has reduced responses to emotional stimuli, right? So for that to be true, of course, you'd have to make a measurement from him and compare him to these populations that you've measured. And suppose we stick him in the scanner and we do the experiment, and his level of response to the emotional faces is that dashed purple line. Well, that's pretty close to the psychopaths, right? So that seems like he's a psychopath, but it also overlaps with the normal controls, right? Now look, this is not anything which is a brand new intellectual problem for anybody in this audience, right? So you're all perfectly familiar with the idea that there might be some things which are true about a general population, which may not be true about an individual, or or the measurement from the individual is not able to completely and with certainty assign them to one group or another, right? This is not anything that's a real shock to anybody in this audience. But it's just worth working through the exercise and seeing that just because this is an amazing brain imaging technique that uses magnets and superconducting wires and measures all this exciting stuff, none of this sort of logical structure is obviated, right? So the same kind of challenges that come to making claims in any other field apply here too. And in fact, I just want to leave us then, and for Oliver to comment upon as well, this even deeper issue, which is if psychopathy is defined as a behavior, how is this neuroimaging data even relevant, right? So if I have a behavioral measure, like a series of pen and paper tests or pen and pencil tests that I use to assign somebody the label of being a psychopath or not, how is getting this imaging data even further inform that definition or that assignment I've had, right? And that's something then which is a bit of a deeper discussion point. So maybe.
Well, before you get there, looking at the last slide, what can you say about probabilities? In
Sure.
other words, what can you say, because judges, we have to look at the individual person. Is it like DNA's probability? I mean, is it 1 out of 1,000,000, 1 out of 1,000,000,000,
Well-
1 out of that? And,
So the question is like, okay, well, look, sure, maybe it's not a certainty, but maybe I could give a very high probability that he fits in one category or the other, right? So wouldn't that be useful information? Sure. I think the point is, though, that to find out what that probability is requires a bit of hard work, which is you'd need to know what is the distribution or range of responses across the psychopaths, and what's the distribution and range of responses across the normal population, and then take his particular measurement and fit it within those continua, right? So that's right. One could do that, and you might come up with an answer like, oh, yeah, it is 99% likely that he comes from the psychopath distribution, right? I think as a general property of this neuroimaging data, to be able to make statements like that, right, and to have some good certainty about the probability, requires imaging not just 10 psychopaths and 10 normal controls, but hundreds. And that's really the challenge of the field in scaling it up.
Oh, you also have to know how certain you are of that. It's not just the degree or the extent of error, or the margin of error, but how certain you are of that margin. Because you might be very, very certain.
That's right.
But you might not be certain.
That's why he's saying the bigger sample size gives you much more certainty about what your error space is.
And broadly speaking, these are solved problems, right? I mean, there's very well articulated statistical methods for knowing about how confident one is of an assignment of an individual to one category or another based upon data like this. It's nothing unique to neuroimaging. It's hard work, and for an awful lot of the sorts of claims made for neuroimaging, that hard work has not yet been done.
Let me, and we've got only a couple minutes left. Let me circle back a little bit, which is to say, what additional work? To follow on, why would we need to know this? Why does this help us? Why does this help us? And one of the reasons it helps us is that at least in theory, although there's some indication that you can spoof the MRI a bit, in theory, this is a physical measure rather than a self-reporting style of measure. That in other words, if you're a smart person and you're doing some kind of behavioral or question and answer style test that may be the thing that gets you your diagnosis to begin with, we've got the malingering problem. Is this person smart enough to know that if I say, sure, I'd kick my brother, or sure, I never loved my mother, whatever the test is, I can end up looking like a psychopath. Now, a more prolonged and careful study of the behavioral stuff can get you better results than that. I'm not saying that it's just that. But one of the things we're longing for in this psychology is that physical confirmation piece. We're just desperate for it in a world where spoofing and malingering is at least one of our concerns. So is this additive in a sense? If it says, by the way, he's 90% consistent, the physical measurements, let's presume they're pretty well developed, are 90% consistent with what we have seen in other folks who we, without having the malingering problem, had defined as a psychopath, well, maybe that gives us that additional layer of confidence in accepting that diagnosis, which we wouldn't have otherwise. So at least a piece of it is that confirmation piece. I'm desperate for some confirmation that psychologist telling me the story in front of me isn't just one of those psychologists who would tell me any story, but is actually, this is good psychology. Here's another piece that does.
You're going to be here for dinner?
I am.
Great.
So I think our time is up. Thank
Our
you
time
all.
is up. Those were great questions. Jeff and Oscar will be here for dinner, and they're available. Let's join me in thanking them.